into place, and it's got its anti-codon next to this codon. So CGC would be GCG. And that's going to come on down and hook up soon, and then it'll form a peptide bond. And then earlier on, if this has been going on a while, a couple things are happening. This messenger RNA code, as it peels off, it's being read, and we've constructed, this is the finished product right here. The messenger RNA strand gets degraded fairly quickly. So it doesn't last long in the cell. So this, I should make a few notes here. So this is called a peptide bond. Peptide bond. And then we mentioned amino acids. We mentioned the transfer RNA. So this thing here is either called a protein or a polypeptide. Don't let that word throw you. It's like poly meaning many, and then these got these peptide bonds holding together. So in the analogy I talked about earlier, this would be lactase, an enzyme that breaks apart lactose. And the information that we got this from was this little small section. This is one of 30,000 different genes found in the nucleus. And the real mystery is, how did it know to go to that part of the DNA and peel it open and transcribe a piece of messenger RNA, and then it leaves and the RNA closes again. And we take this little fiber here of information and we make this. And this goes about, this new protein will go about breaking apart lactose sugar. And also miraculous is when we're done, when the job is done and all the lactose sugar that you've consumed is now broken by lactase into the two simple sugars, even the protein gets disassembled and those amino acids are ready to be used a different time in a different way. So there's a lot of cool mystery about this, but this is the process. And by the way, this whole building part is called translation. The reading part is called transcription, so I could put an arrow over here. So this is chapter 17. This is called protein synthesis. And so you can see that the DNA never leaves the nucleus. The information just stays there. And when you need something, you go in there and you peel it open, and you can make an RNA copy of the DNA. And there are some exceptions, in terms of, I think of the word unity, U in place of T, U not T, unity. Anyway, so these RNA letters will never have a T associated with them, but you will see a U every now and then that replaces that. So anyway, this is the picture of how things get built. And there's a lot of mysteries. How does it know where to find that particular thing? And then, how many replications do you get? How many proteins do you build with one messenger RNA molecule? Or can this be going on multiple places, multiple times? And the answer is yes, there's a lot of flexibility to those things. So anyway, if this makes sense, then you've kind of got that notion of protein synthesis down. Cool.